Hello students, welcome to the lecture on hearing. So the ear detects sound, it detects head movement. We're going to use cranial nerve number 8, the vestibular cochlear nerve, to convey both of those types of sensory signals. The external ear has an auricle that you can see over here. That's the big funnel-shaped part of the outer ear. It's got elastic cartilage, so it springs back to its original shape if you deform it. It protects the ear, directs sound into the external acoustic meatus, which is your ear hole basically. It goes all the way from the outside to the tympanic membrane. So all of that is going to be the external ear. The tympanic membrane is a funnel-shaped sheet of epithelial tissue that separates the internal and external middle ear, and it vibrates when sound hits it. Now you've got air inside that external acoustic meatus, possibly earwax or small bugs or something. But inside the middle ear, you've got air. There's no ceruminous glands that are present inside of the middle ear. The air is continuous with the back of the throat through the pharyngotympanic tube, also known as the auditory tube, or it used to be called the eustachian tube. And that open passageway allows us to equalize the pressure between the outside atmosphere and our middle ear. If you yawn or chew gum or open your mouth up really wide, that tends to open up that passageway if it's a little bit stuck, and that helps to equalize the pressure. If it gets plugged, then as you change outside pressures, the inside doesn't change, and it will cause the tympanic membrane to deform and to either bend into or out of the skull, causing a great deal of discomfort either way. Inside the middle ear, we've got three tiny little bones called the malleus, the incus, and the stapes. The malleus is a little hammer. The incus is an anvil, and the stapes is a stirrup. Here's a life-size picture of just how tiny these little bones actually are. These little tiny bones function to magnify sound waves. As vibrations from the outer atmosphere move the tympanic membrane, that causes the malleus to start to move, and that's going to cause the movement to go to the incus, which is then transferred to the stapes, which kind of punches in and out of the oval window. And as it does so, it's going to cause some pressure waves. Just as if you gently put your hand on top of the surface of water in a bathtub and started to move your hand up and down, not a lot, but just to start making ripples. That's what the stapes is going to do, is it's just going to kind of punch in and out and cause ripples in fluid that we're going to have inside the inner ear. There's two very small muscles I want to look at here. There is the tensor tympani muscle, which attaches to the malleus, and the stapedius muscle, which attaches to the stapes. Both of these muscles will function in pulling these little bones out of alignment during sudden, loud noises to prevent damage from occurring to the inner ear. Now, because these muscles are so small, they're going to fatigue very, very quickly. In fact, within about 3 seconds, they just kind of give up. The bones slide back into place and continue to magnify the sound. So loud sounds longer than 3 seconds or so are going to do more significant damage to the inner ear. The inner ear has spaces within the petrous portion of the temporal bone. There's a bony labyrinth, which is a maze-like space inside of the bone, and there's fluid that fills up that space called perilymph. And then also inside of there, there's a very long, tortuous membrane sac called the membranous labyrinth, and that contains a slightly different fluid called endolymph. Perilymph is higher in sodium, endolymph is higher in potassium. Here we can see the structure of the inner ear. The cochlea is our snail. It houses the membranous cochlear duct. The vestibule is the entranceway into the inner ear. Here's the oval window. The vestibule contains two sac-like membranous parts, the utricle and the saccule. They're interconnected, and they are used to sense static positioning and linear acceleration of the head. And then finally over here we have our semicircular canals. These contain semicircular ducts, and they are used to sense dynamic equilibrium and rotation of the head. Here's a cross-section of the cochlea. Remember, cochlea means snail in Latin. The medullus is that bony axis of the spiral, and then the cochlear duct wraps all the way around that medullus. Here's a close-up of the cochlear duct. Remember, the vestibular membrane is the roof, and the basal membrane is the floor, so this pinkish area here is actually our cochlear duct. Cochlear duct is the membranous part of the labyrinth, whereas the scala vestibuli and the scala tympani are the bony part of the. As our little snail winds around the medullus, the very tip of it is called the helicotrema. There's a small channel in there that connects the scala vestibuli to the scala tympani. The spiral organ of Corti is the sensory structure we use to do hearing. This is going to be on the basilar membrane, or the floor of our cochlear duct. There is a kind of a thick sensory epithelial layer here that consists of little hair cells and supporting cells that sit on top of that basilar membrane. The hair cells contain receptors that are going to release neurotransmitters. We have a single row of inner hair cells and 3 rows of outer hair cells. The hair cells contain little specialized long microvilli called stereocilia, and one especially long cilium at the very apex called the kinocilium. The stereocilia and the kinocilium are embedded within the tectoral membrane. So the base of these little hair cells is in the basilar membrane. The kinocilium and the stereocilia poke up, and the very tips of these are then embedded within the tectoral membrane. So as the tectoral membrane moves a little bit, it's almost as if you took a hairbrush and pushed it into a soft bar of soap just a little bit. You can clearly see the bristles, but then at the tips of the bristles, we've got that kind of bar of soap stuck in there. So that bar of soap would be the tectoral membrane, the bristles would be the microvilli, and then the body of the brush would be the basilar membrane. Now the base of the hair cells are going to synapse with our sensory neurons. As sound waves in the outer atmosphere cause movement of the tympanic membrane, that movement or vibration is going to be picked up by the 3 ossicles and magnified as it's transmitted to the oval window. That's going to cause a wave of fluid pressure as that stapes goes in and out of the oval window, and that fluid wave is going to travel through the scala vestibuli, and as it does so, it's going to either push that vestibular membrane down a little bit, or it's going to suck it up just a little bit. As that occurs, that's going to cause the endolymph inside of the cochlear duct to follow, causing the basilar membrane to be pulled up just a little bit. That change in pressure and the bending of the basilar membrane is then going to cause hair cells to distort, and that's going to cause changes in neurotransmitter release on the vestibular cochlear nerve. Specific regions of the basilar membrane are going to move more than others depending on the frequency of sound. The pressure wave is then transmitted down through the basilar membrane to the scala tympani, and it's absorbed by the round window. As we look at the sound pathway as it moves through the scala vestibuli and the scala tympani on the way back out, you'll notice that we're going to go in here through the oval window, through the scala vestibuli, all the way around until it hits a certain portion of that cochlear duct where it's going to cause the most deformation of the hair cells and therefore the largest change in neurotransmitter release. In other words, it's going to stimulate the nerve at that particular spot. Any excess pressure is going to continue through the scala vestibuli all the way to the helicotrema and then through the scala tympani to the round window. And that system's in place just to help balance out the pressures. Now this is really nifty. The inner hair cells contain ion channels at their tips, and there's tip link proteins that connect them. The hair cells are bathed in endolymph. Remember, endolymph is what we find inside of the cochlear duct, and it's high in potassium. When the basilar membrane moves up, the hair cells are pushed into the tectoral membrane, and their tips are tilted, pulling up the tip links. The tip links will then open their ion channels and allow potassium to diffuse into the hair cell, and that starts to depolarize it. The hair cells release more neurotransmitter from its base, exciting the sensory neuron, which then fires action potentials. When the basilar membrane moves down, the process reverses. Now, hearing is the perception of sound, and sound is just a pressure wave in the atmosphere. Everything in the universe tends to vibrate. Vibration pushes molecules, which then transfer energy from one molecule to the next, kind of like billiard balls. Now, pitch depends on the frequency of how fast the object is vibrating, and we measure that in cycles per second, also called a hertz. Humans can detect anywhere from about 20 cycles per second to 20,000 cycles per second. Variations in pitch are detectable due to variations in the stiffness of the basilar membrane from the oval window to the cochlear apex. In other words, as we move from the oval window to the cochlear apex at the helicotrema, the membrane becomes less stiff. This causes high frequency sounds to excite the cells in the stiffer basilar membrane near the oval window, and lower frequency sounds to excite the cells in the more flexible basilar membrane near the apex. So here's a sound wave. Frequency is the number of times that a wave passes a given point. Wavelength is how long the wave is, or the distance between two consecutive crests, and amplitude is the height of the crests. Loudness is dependent on wave amplitude, how high the wave is. Pitch is determined on the wavelength or frequency. The more frequent the waves are, the higher the pitch. The higher the waves are, the louder the pitch. We measure loudness in decibels. So in this example here, this line is happening at twice the frequency of this line. That means that the first line is going to have a higher pitch than the second line. Here the two lines have the same frequency, so they'll have the same pitch, but this line here is going to be louder, and this line here is going to be soft. Now, fibers that span the width of the basilar membrane are short and stiff near the oval window. They resonate in response to high frequency pressure waves. The longer fibers near the apex that are part of that more flexible basilar membrane are going to respond to lower frequency waves. This is actually really good. If you've ever been in traffic and somebody's been kind enough to share their music with you from 4 car lengths away, it's only the bass that you hear, because those longer frequency waves tend to penetrate a lot of substances, including metal. Higher frequencies tend to be stopped fairly easily, and so as we're looking at the length of the cochlear membrane, we want to capture those high frequencies as fast as we can before they get blocked by anything. Whereas with the longer frequencies, we can let them go to the very end where they'll finally start to stimulate that basilar membrane.